Good evening, everyone. I'm Jyoti Sharma. But before starting this, I just want to let me know whether my voice is audible to you or not. Can someone reply whether my voice is audible to you or not? Okay, Naveen, thank you so much. Okay, so let me start once again. I'm Jyoti Sharma, and this is week 10. And the content that we'll cover today will be from lecture 49 to lecture 53. And basically, we have a lot of MCQ question on bacterial growth curve, and we have monad kinetics, a law of the minimum. Then we will talk something about how we can culture the bacteria and what are the instruction that we should take care of while culturing the bacteria and the different technique, for example, plate counts and membrane filtration, microscopic method. Then we will talk about something about the factor that will affect the microbial growth. Then we have some terminology, for example, chemostat, batch culture, heat sterilization, disinfection. We will talk about such kind of terms. And then yes, a different way by which we can kill or inhibit the growth of the microorganism, for example, using the radiation or using the filter sterilization, or we may have antimicrobial activity and disinfection. So we will talk much about this. But okay, so let me start with my first question. And this first question is on the curve of the growth of the bacteria. So, my question is, during which phase do bacteria divide at the fastest possible rate? So, if you have seen the bacterial growth curve, then we have four phases. That four phases are lag phase, log phase, stationary phase, and death phase. So, you have to tell me in what phase bacteria divide at the fastest possible rate. You can write your answer in the chat box. Even if you are giving me the wrong answer, don't worry about this because I will correct you. So, what kind of phase I'm talking about when bacteria divide at the fastest possible rate? Is anyone interested to give me the answer for this? You just have to tell me the phase of bacteria when they divide at the fastest possible rate. Okay, so the answer is log phase, and let me tell you why it is the log phase. Then just see that in the log phase, this is the fastest, and why this is fastest, because the rate with which the bacteria grows is exponential rate. And let me show you the graph of this. So this is the bacterial growth curve. So just see the first portion. Let me take the pen. Okay, so this first portion is known as the lag phase. So this is the lag phase when there is no any increment in the number of the bacterial cell. But what will happen then? Another phase. This is the log phase when we do have the exponential growth of the bacteria, and so this is one of the fastest. Growth rate of the bacteria. Then we have the stationary phase, and in this stationary phase, the number of bacteria are growing will be the number of the bacteria that are dying. So this is something known as the stationary phase. And then we have the death phase, because um, uh, after consumption of food, after consumption of like whatever the material that has been provided, that material will be insufficient for the bacteria to grow. And so that what will happen, the death of the bacteria will be more as comparison to the growth of the bacteria. So this will lead to the decline phase, or you can say the death phase. So these are four phases we have, and the question was on the fastest growth rate. So this fastest growth rate will be in the log phase. So that's why this log one answer is right one. Okay, the next question is, what is happening when bacterial cell prepare for division but show no immediate increase in number? So again, you have to tell me the phase, and the phase you have to like guess the phase when bacteria prepare for the division uh, but doesn't divide. So my four options are: they are in the log phase, they are undergoing exponential growth, they are in lag phase, and they are entering the death phase. Let me show you the curve again. So this is our curve: lag phase, log phase, stationary phase, and the death phase. So you have to tell me the answer based on this phase. And my question is, when bacterial cell prepare for division but show no immediate increase in number, so what kind of phase I'm talking about? Can you guess? Okay, so this is lag phase. So let me tell you. So let me take the pen. Okay, so this is the lag phase, and this lag phase is something when bacteria adapt to the climate, when bacteria have the nutrient in available amount, but bacteria doesn't divide and doesn't give you the exponential increment. But this is the time when bacteria is having the adjustment with the climate, adjustment with the nutrients, adjustment with the food that has been provided to the bacteria. So this is the phase when you will see no increment in the number of the bacteria, but this is the time when they are adapting to the climate. So in this lag phase, these bacteria do not divide, but they are adapting with the climate, but they also have the sufficient amount of nutrition, or you can say the food. So this is lag phase. So the answer will be they are in a lag phase. So lag phase is the adjustment period where bacteria synthesize necessary enzymes and adapt to new environmental condition before beginning rapid division. So I think this is clear now why this is lag phase, not the log, not the stationary, and not the death phase. This is again, you can see, so this is for the growth, and this is for the substrate, or you can say the food. So food will decrease when the number of bacteria will increase, and there will be one time when we do not have like sufficient food, then this will be the stationary, and after consumption of 100% food, then there will be the death phase. So this is something, the graph between the concentration of the bacteria versus substrate. Okay, the next question is, which phase is characterized by an equal rate of growth rate and death? Again, the question is based on a simple bacterial growth curve, and the options are lag phase, log phase, stationary phase, and death phase. So, can you write the answer in the chat box? When the death rate is equal to the growth rate. So, this is kind of equal number of growth to the equal number of deaths. So, what kind of phase I'm talking about? Can you give me the answer in the chat box? Anyone? Let me again show you the curve. So this is the curve. We have lag phase, we have log phase, we have stationary, and then we have the death phase. So my question is, when there is a equal rate of bacterial growth and death, so what kind of phase I'm talking about? So this is the stationary phase. uh Not log phase. Log phase is the exponential growth. So exponential growth means there will be very less death as comparison to more growth of the bacteria. So this will not be the log is exponential growth. So exponential means there will be very less death of the bacteria as comparison to the growth of the bacteria. That's why it is moving upward. And the stationary is like this. So node like the number of death will be equal to the growth of the bacteria. So this should be the stationary phase. Is it clear, Gyanendra? That this will be stationary phase only. So in stationary phase, nutrition depletion and waste accumulation prevent further net growth, and that will balance cell division and death. Let me again show you. This is the case. So this is the stationary phase, and this stationary phase means the number of death will be equal to number of growth of the bacteria. Thank you. So let me come to the, okay. So this is the numerical question, and very, very simple numerical question. So let's say your bacteria culture starts with 50 cell and grows to 32 100 cell in 5 hours. If the cell divide at the constant rate, how many generation have occurred? And my options are 4, 5, 6, 7, and we are considering it the logarithm growth of the bacteria. Logarithm growth means or you can say the exponential growth. So initially you had the 50 cell, then 50 cell will divide to form the 100 cell, 100 cell will divide to form the 200 cell, 200 cell will divide to form the 400 cell. So you have to tell me how many generation has occurred till the number of cell is 3200. And in 5 hours. So 100 to sorry 50 to 100, 100 to 200, 200 to 400, 400 to 800, 800 to 1600, 1600 to 3200. And you just have to tell me how many generation we have. Can you calculate with this exponential growth? So the answer is 6. So just see, no, 50 to 100, 1, 100 to 200, 2, 200 to 400, 3, 400 to 800, 4, 800 to 1600, 5, and 1600 to 5200, 6. So these were the 6 generation from 50 cell to 3200 cells, right? So this is how you can calculate. And we have one formula as well. So this is n node. N node is a number of cell you had initially, multiply by 2n, and this n is the number of generation. And whenever you will solve this, whenever you will find this n, then you will get that this number of generation is 6. Okay, the next question is, a bacterial culture has gone through 4 generations, starting with 5 cells. So, how many bacteria are present now? And I told you this is the exponential growth. Can you tell? Like we started from 5 cell, and after fourth generation, how many bacteria we will have? 5 to 10, 10 to 50, 50 to 100, 100 to 200. So we have four generation. Can you tell me after fourth generation how many bacterial cell we will have? 20, 40, 80, 160. Very simple. 5, 5 to 10, one generation. 10 to 20, second generation. 20 to 40, third generation. 40 to 80, fourth generation. So 80, because we will have the fourth generation. So, and here again, we do have very simple formula again. So n is the number of cell, is equal to number of cell initially we had, multiply by 2 raised to power n, and here n is the number of generation, and the n is given a 4, so number of generation were 4. Initially the number of bacterial cell were 5, so put every value and you will get 80. So 5 multiplied by 2 raised to power 4, and this will be 5 multiplied by 16 is equal to 80. Is it clear why the answer is 80? Okay, so let me go to the next question. This is again very similar question, and I'm expecting the answer from you guys. So the question is, a bacterial population doubles every 30 minutes, and if a culture starts with 200 cell, how many will there be in 2 hours? So first you have to tell me how many generation has been completed after 2 hours. So this will be, and because one generation take 30 minutes, right? And in 2 hours how many generation you have, and then based on the number of generation, the initial number of cell, you can calculate the bacterial cell after 2 hours, right? So the number of generation will be, because there is 30 minutes, right? So 30 minute, 60 minute, then 120 minute, and then 180 minute, is it? No, this is not like this. So this is 30 minute, 60 minute, 120 minute, and okay, you people are not cooperating with me, but let me tell you. So initially we have 30 minute, then after 60 minute we had second generation, then after 90 minute we had third generation, and after 120 minute we had fourth generation, and 120 minute means 2 hours. So total we had four generation. Okay, so n value will be 4, and then n node, so number of cell that we had initially is equal to 200 cells. So what you need to just calculate is n. So this is the present number of cell, so this n will be equal to, let me tell you, so this n is equal to n node and multiply by 2n, and this n is the number of generation, and number of generation is from 30 minute to 2 hour, we have total four number of generation, right? And n node is equal to 200. So now can you tell me the number of bacterial cell that we will have in 2 hours? So this will be 1600, and let me show you how. So first, as I told you, first you calculated the four generation that we have, because we started with 30 minute, 30 to 60, 60 to 90, and 90 to 120, right? So, and this is 2 hour. So after 2 hour we have 1, 2, 3, uh, 1, 2, 3, and 4. So we have four generation, right? And after that we will put this into this formula, 200 multiplied by 2 raised to power 4, and this is how we will get the 1600 bacterial cell that we will have after 2 hours. Okay, the next question is about the plate counting essay. And the question is, what might bacterial colonies fail to appear after performing a after a plate count assay? And the options are: bacterial cell formed colonies too small to see, the incubation temperature was optimal, the medium provided sufficient nutrients, and the culture contained no viable cell. So when you will fail to see or to observe the bacterial colony after performing a plate count assay. So just see here, uh, it is not possible that if we have the colony we can't we can't see the cell. So this is the wrong statement. Whatever the smaller colony we have, we can see that. So this is not the right answer. Then if the incubation temperature is optimal, then this is giving us the conducive environment to grow the bacteria and to see the bacterial colony. So this is again not the right option. Then medium provided sufficient nutrients, and if the medium is providing the sufficient nutrient to the bacteria, then there must be sufficient colonies to grow, and this is how this is again not the right option. Yes, Ganendra. So D is the right answer, that uh, culture contained no viable cell. So if there is a no viable cell, then there is a high chance that you will not observe any bacterial colonies. So that's why D is the right answer. So colonies may not appear if there were no viable bacteria in the original culture. Other possibility may include improper incubation condition or unsuitable media. But if cells were completely absent, no colony will form at all. So that's why D is the right answer. And here you can see, initially you had this original sample, then you did the dilution, then you take the 1 ml out of this 10 ml, and you add 9 ml of the other solution, and this is how you do the dilution of your culture. And then initially in the plate, let's say you had a lot of bacterial cell, but after dilution you will have the minimum, and that will give you the easier estimation of the count of the bacterial cell. So this is how we can grow the colony, and we can see after the dilution we can easily count the number of bacterial cell. Okay, so the next question is, a researcher perform a plate count. So plate count is nothing. So on the plate you have the uh, you have agar, let's say, and on that agar you have some colonies of the bacteria, and you need to count that colonies of the bacteria. So this is something about the plate count. So if a researcher performs a plate count but sees no colonies after incubation, which of the following is not a list cause? So just see here, this is a trickier question because here there is a not a likely cause. Then options are: bacterial strain requires spatial growth factor not present in the medium, the culture was heavily diluted to a point where no cell were plated, bacterial cell were present but did not divide because they were in the lag phase, at incubation temperature was not suitable for growth. If you are not seeing any colony, so just first see what can be the cause of it, and out of that absurd cause you will get not a likely cause. So what will be the right answer for this? So this is bacterial cell were present but did not divide because they were in lag phase. Let me tell you that this is the reasoning here. The lag phase is the period of adjustment, as I already told you, but it doesn't prevent cell viability. So it doesn't mean that bacterial cell will not be the visible, right? So it will just will not multiply, but whatever initial bacterial cell we had, these cell must be observed, and if this is not observed, then the reason is different, but it is not the reason that they are in the lag phase. Okay, let me come to the next question. A scientist perform a plate count using the dilution series. As I told you about the dilution, initially we had, let's say, 10 ml 10 ml of the solution having the bacteria, then we will take the 1 ml and then we will add something other in that 1 ml again. Out of this we will extract out 1 ml, and this is known as the dilution series. And a scientist perform a plate count using a dilution series but only see colonies on higher concentration plate. What does it suggest? So we have dilution. So for example, initially we had 10 ml of the solution, then out of that we took 1 ml, then out of this we took 1 ml, out of this we took 1 ml and diluted with some other solution, right? So what will happen, the number of the bacterial cell that we had in the first stage will be higher as comparison to the bacterial cell that we will have in the dilution series. So if you are not able to see uh, higher number of the colonies in the coming dilution series, so what does it suggest? So my options are: the initial culture contained uh, too few bacteria for lower dilution to show growth. The bacteria were were no viable in the original sample. The incubation temperature was too high for growth, and the bacteria contained an antibiotic that killed the bacteria. Right now we are talking about the dilution. With the dilution, the number of the number of bacterial cell will be reduced because we are taking smaller amount of the solution that had the bacteria, right? And if we are not able to see the bacteria in incoming dilution series, so what does it mean? So it means that in initial culture we had very few bacterias. Okay, so let's say in first test tube we had just 50 bacteria, and then out of this 10 ml of the bacterial cell, what we are taking, we are just taking 5 ml of this, and we are filling it with the 9 ml other solution. So we have just 1 ml that had the bacteria. So this 1 ml will may have, let's say, 25 bacteria. Then what we are taking out of this, we are again taking 1 ml of the solution. So this is how it may be that in the coming dilution series we may not able to see the bacterial cell, right? Because in the initial the number of bacterial cell were low, these were not very highly, or will not had not very concentrated in nature. So this is how, yes, A is the right answer. So if colonies appear only on plate with higher concentration, it suggests the initial culture contained very few bacteria, making it unlikely for the lower dilution plate to receive viable cell. If all bacteria were non-viable, no colonies would appear at all, right? The other option could also prevent growth but would typically impact all plates equally. So that's why A is the right answer. And this is how what I was trying to tell you, that initially we had 1 ml of the solution. Then what we do, out of this total 10 ml we took 1 ml, and out of this 10 ml again we took 1 ml, and this is how the colonies, the number of bacterial colonies will reduce to, let's say, 0. And for example, in initially we had a very few colonies, then in the coming dilution series we will get nothing. Okay, the next question is, which of the following statement about the plate count method is false? And we have four options. It provides a quantitative estimate of viable bacterial cell. It can distinguish between living and dead cell. It requires incubation for bacteria colonies to appear. It involves plating a known volume of a diluted bacterial culture. What do you think? What is the right answer for this? The false statement about the plate count method. So, so the wrong statement is B, that this plate count method is not able to differentiate between living and dead cell, and that's why if it is distinguishing between living and dead cell, this is the wrong statement, because plate count method is not able to differentiate between living and dead cell. So that's why B is the right answer. Okay, the next question is, a researcher wants to count bacteria in a sample immediately without waiting for incubation. Which method should they choose instead of a plate count method? So because for the plate count method we need to have some time so that the colony can be grow, but if we do not have much time for this incubation, so what kind of method we can adopt instead of this plate count method? So what we can do is we can use direct microscope count, right, in spite of growing the colonies. So this is, and can you tell me the example of direct microscopic count, because you have to count, so you have that kind of a slide in which there is a the squares, and in that square you can count the number of cells, and you do not need to grow the colonies, and if we are growing the colony, this will come under the plate count method, and if this is not the plate count method, then you can directly rely on this direct microscopic count. And one of this method can be this may be hemocytometer or petrophosphor counting chamber, and here you can see we have the slide, and in that slide we have lines, and in that lines we can count the microbial cells. So if we do not want to go for this plate count method, you can just see the bacterial cell under direct microscopic, and you can count the number of bacterial cell. And again we do have the limitation of this, again we cannot differentiate between live and the dead cell using this method. The next question is, a food scientist perform a plate count on a juice sample and observe far few colonies than expected. Which of the following is the least likely explanation? So again this is just like that, we what we are observing very less colonies on the juice sample, and you have to tell me the least likely explanation for this. And the options are: the bacteria in the sample were in the viable but non-culturable state; the agar medium used did not support the growth of all bacterial species; then bacteria in the species were mostly dead; the juice contained a high concentration of nutrients promoting rapid growth rate. And you have to tell me the least likely explanation. So the least likely explanation is a juice contain high concentration of nutrient promoting rapid growth rate. So for example, if you have high concentration of nutrient, then it should give us the uh, larger colonies than expected, but the question is it is giving you the fewer colony than expected. So this is absurd statement, and that's why this is the least likely explanation for the fewer colonies than expected. So that's why answer D1 is the right one. Okay, so next, coming to the next question, this explanation I have given already. Okay, which of the following best explain why bacterial cell do not immediately start dividing when introduced into a new environment? As I told you, we had the lag phase, then log phase, then stationary phase, and then the death phase. And in the lag phase, this lag phase is the time when the bacterial cell will not divide, but will take some time to adapt with the climate. So and the question is just like only, and we have four options. The cells are undergoing DNA replication before division, the cells are synthesizing essential enzyme and adapting to new condition, the cells are being outcompeted by other organism, and D is the cells are in dormant stage with no metabolic activity. What do you think, what is the right answer? In the lag phase, before multiplying exponentially, bacteria takes time before the division of the cell, and this is because they are adapting to the new condition or to the new environment, and that's why in this time they are not expending much energy on the reproduction, but they are giving this energy to to synthesize essential enzymes. So that's why B is the right answer. And just see here, During the lag phase, bacteria do not divide immediately because they are busy adapting to the new environment, synthesizing necessary enzyme and preparing for growth. So that's why this is the right answer. B is the right answer. Okay, then again, we have question on the bacterial growth curve only. I think we have last second question on this, and then we will move to the another topic. So question is, a bacterial culture is introduced into a fresh medium. Okay, but no increase in population is observed for several hours. Which of the following is the least likely explanation? And we have four options. The bacteria are synthesizing new enzyme for the metabolism. The bacteria are in the stationary phase. The bacteria are adjusting to the new nutrient condition, and bacteria are repairing cellular damage before division. And the question is, which one is the least likely explanation? So if there is a no increment in the population for several hours, so what is the least likely explanation? So if there is a no increment in population, as I told you, they must be in the lag phase, and lag phase means they are synthesizing new enzyme. So this is not the least likely explanation, this is the likely explanation. Then bacteria are in stationary phase. We will talk about it. Then bacteria are adjusting to the new environment condition. This is again the likely explanation, but the question is least likely explanation. And bacteria are repairing cellular damage before division. This is again the likely explanation, but not the least. So the remaining one is the stationary phase, because whenever we have the bacterial culture in the fresh medium, so this will come under lag phase, not the stationary phase. So that's why B will be the right answer for this. Is it? Okay. So yes. So let me give you the another question. If the bacteria in lag phase are transferred to the fresh medium with the same composition, what is most likely to happen? So let's say the bacteria were in lag phase in some other another medium, but what we are doing, we are transferring this number of the cells into the another medium, but that another medium also have the same composition. So what will happen to the bacterial cell? So I have four options. The lag phase will restart and bacteria will take time to adjust. The bacteria will immediately enter the log phase and start dividing. The bacteria will die due to sudden environmental change. The bacteria will enter the stationary phase earlier than expected. So the condition is only that we had a fresh medium, and that in fresh medium the bacteria were in lag phase, but what we did is we transferred this bacteria into the another medium, and that another medium also had the same composition. So what will happen, what kind of impact it will give on the growth of the bacterial cell? So the answer is B, that the bacteria will immediately enter the log phase and start dividing, because there will be the same composition, same environment. So they already have adapted to the same environment, adapted to the same composition of the nutrient. So there is no any way that it will restart the lag phase, but it will start from where it has been stopped. So after the lag phase there will be the log phase. So B is the right answer. Yes. So if bacteria are transferred to the identical fresh medium, they are already adapted to the condition and do not need to undergo another phase, another lag phase. They immediately enter the log phase and start dividing. So that's why this answer is the right one. Okay, the next question is, what happens first in the binary fission process? As we already have discussed, then in the bacteria, they do not have the sexual reproduction, but this is the part of asexual reproduction where the cell divide into two using the method, and that method is the binary fission. So you have to tell me what happens first in the binary fission process. So we have four options, and this is invagination of the plasma membrane, B is the formation of the septum, C is the replication of the bacterial chromosome, and D is the complete separation of the daughter cell. So first is the replication, right? So initially you had some chromosome, and there will be the replication. After that replication, the cell membrane will start come closer, will differentiate these two chromosome, and there will be a septum that will give you the two number of bacterial cell. So the first step is the replication of the bacterial chromosome. Let me show you how. So initially we had this is their genetic material. This genetic material first will replicate. After this replication, the cytoplasmic membrane will elongate it, and this will try to make a septum, right? And then they will stretch out and will give rise to the two daughter cell. And so the first step is the replication of the DNA. Okay, or you can say the replication of the chromosomal DNA. Okay, coming to the next question. If a scientist is culturing bacteria and find out that microorganism or organism can grow in the complete absence of oxygen, so the microorganism does not need the oxygen, so which of the following statement is most accurate? The bacteria must be obligate aerobics. The second is bacteria may use alternative electron acceptor like nitrate or sulfate. The bacteria will die because oxygen is essential for metabolism. And D is the bacteria must be photosynthetic. What do you think? What is the most appropriate statement when we are saying that bacteria or the organism doesn't need oxygen to grow, and the organism can grow in the complete absence of oxygen? Can you give me the right answer for this? Okay, so let me show you these options and the clarity about the options. So the first is the bacteria must be obligate aerobics. It is not it is not always correct that the bacteria must be compulsory aerobic in nature, or the bacteria that needs the oxygen. This is not the right statement, because some of the bacteria are here that do not need the oxygen, so they can completely grow in the absence of oxygen. So must be obligate aerobic, this is not the right statement. Then bacteria may use alternative electron acceptor like nitrogen and sulfate, this is right. Then bacteria will die because oxygen is essential for the metabolism. No, the statement is like the organism can grow in the complete absence of oxygen. If they can grow, then how this is oxygen is important for the metabolism? So this is wrong statement. Then bacteria must be photosynthesis. No, this is again not right, because if they can grow in complete absence of the oxygen, then it means they are not doing this photosynthesis, and they can even survive without photosynthesis activity. So this is again the wrong statement, that bacteria must be photosynthetic. So the only option that we have is the B option. So B option is the right one. So if you see here, we have the electron tower, and if we do have the anaerobic and anoxic condition, then the sulfate, nitrate, iron, CO2, hydrogen can replace this oxygen, right? Okay, coming to the next question. A researcher finds that a bacterial species require nitrogen but cannot use atmospheric nitrogen. What is the most likely explanation? And the options are: the bacteria lack nitrogenase enzyme, the bacteria do not need nitrogen for growth, the bacteria can directly use N2 without conversion, the bacteria are capable of fixing nitrogen, atmospheric nitrogen. So what do you think? When bacteria is not able to take or not able to use atmospheric nitrogen, so what can be the possible best explanation? So the best explanation is because that bacteria do not have that enzyme. That enzyme can fix this nitrogen, atmospheric nitrogen, to transform into, let's say, ammonia. So they do not have that particular enzyme, so that's why they cannot directly use this atmospheric nitrogen. Okay, so A is the right answer, that bacteria lack nitrogenase enzyme. Then bacteria do not nitrogen for growth. This is the wrong statement because, like, to my best knowledge, all bacteria need nitrogen for the growth. Then bacteria can use nitrogen without conversion. No, this is not the option, because whatever organism we are talking about, most of the bacteria and in another eukaryotes and the prokaryotes, we need nitrogen, and some of the bacteria can uh, transform this N2 into ammonia stage. But if they are not able to do this, that means they are lacking this. But what they have to do is they have to take it from somewhere else, but they can't directly use this N2. So this is not the right statement. And bacteria are capable of fixing atmospheric nitrogen. For this atmospheric nitrogen fixation, they need the enzyme, and if they do not have the enzyme, they can't use this atmospheric nitrogen. So this is again the wrong statement. The right statement is only A. Yes, this is A. So some bacteria can use atmospheric nitrogen directly, but may lack the enzyme nitrogenase, which is required for nitrogen fixation. But these bacteria rely on preformed nitrogen source, like ammonia or nitrate. That's why that A answer is the right one. Yes, Yuki gave me right answer. So yes, he is the right answer. Okay, the next question is, again we have a numerical question, and here we have bacteria culture start with 5 cell and divide every 20 minutes. How many bacteria will present after 1 hour? So first, what you have to do is you have to calculate the number of generation. After that number of generation, you can calculate the number of cells that we have after 1 hour. So options are 20, 40, 80, and 40 multiplied by 2 raised to power 3. So okay, so first calculate the number of generations. So it started with 20 hours. So after 20 hours, so 20 hours to 40 hours. Sorry, 20 minutes to 40 minute, it will have the first generation, right? Then 40 to 80, it is the second generation, but it is 60. Then this is 1.5 generation, and this, if this is 1.5 generation, then you have to tell me the number of cells that will we have after 1.5 generation. But the number of cell initially we had 5 only. So can you tell me the number of bacterial cell that we have after 1 hour? So how you can do this? So let me show you. uh First is, okay, so here I did the mistake that I didn't tell you the number of generation is equal to 3, because 20 20 20. So this is how this is 60 minute, and 60 minute means we have 3 generations. So what I was doing just right now is the exponential growth, but for the time we just have to see the multiple. So 20 minute 20 minute 20 minute, so this is giving us the 3 generation, right? So n will be equal to 3. Then you have to have the number of initial cells. So initial cell we had is equal to 5, multiply by 2 raised to power n is the number of generation. So n here is equal to 3, and this is 5 multiplied by 2 raised to power 3, and this will be 5 raised to power 5 multiplied by 8 is equal to 40. So this is how we will have the 40 number of cells after 1 hour, right? So the mistake I did is, uh, I was doing like 20 to 40, 40 to 60, but this is about the exponential growth of the bacteria. But if I'm having the timing, the double timing is just the 20 minutes. So 20, then whatever the bacterial cell I have after one generation, this will be double after 20 minutes. So 20, 20, 20, this is how it will give me the three generation, right? So this is how the answer is 40. Okay, so again the answer, again the question is, the bacterial colony start with 1000 cell. If the bacteria double every 40 minutes, how many cells will be present after 2 hours? So here the double time is 40 minute. So 40 minute, then 40 minute, then 40 minute, 40 minute, and you just have to see when you will have the 2 hours. So this will be 80 minute, this will be 120 minute. So this much generation only, so this much generation only will give you the 2 hours. And then it started with the 1000. So after 1000 it will be 2000, after 2000 it will be 4000, and after 4000, let's say, it will be 8000, right? So this is how you can solve this question. So just see here how we can solve this. So first we calculated the number of generation. So in 2 hours we had 120 minute, and the doubling time is 40. So the number of generation will be 3, right? And then what we have to do is initial count multiply by 2 raised to power n, then initial count over 1000, and then number of generation is 3. And this is how 1000 multiplied by 8, this will give us the 8000 number of cells after 2 hours, when the doubling time is 40 minutes. Okay, coming to the next question. Okay, next question is based on the medium. So we have a different type of medium to grow for the bacterial culture, and this question is on this different type of medium. So what kind of medium we have based on sub prescription or based on some characteristics. So the question is, a scientist is culturing a new discovered bacterium and wants precise control over nutrient concentration. Which type of medium should be used? So we have four options: complex medium, defined medium, enriched medium, or selective medium. Can you give me the answer, Yogi? Yanindra, can you give me the answer for this? So when we need a very precise concentration of the nutrient in that culture, so what kind of medium we are talking about? So we are talking about the not selective, Canindra. This is defined, and so defined have exactly, we know the chemical composition of the uh, medium. Then this will come under this defined medium. It could have been the selective medium, but why this is not selective? A selective medium is used when we just want to grow, let's say, some specific type of bacteria, and we are removing another type of bacteria. So if that would be the condition, then we would have been used this selective medium. But here what we want is, we want precise control over nutrient concentration. So if this is the exact, we if we want to know the exact composition of the nutrient that we are adding into the culture, then this will come under defined medium. So B is the right answer. And just see here, a defined medium contain precisely measured chemical component, allowing the scientist to control nutrient level. And if we are talking about the complex media, then it contain undefined ingredient like peptone or yeast extract. If we are talking about the enriched medium, then these have very special nutrients that can give you the like more growth of the bacteria, and selective media favor the growth of certain microbes by inhibiting others. So this is how the B answer is the right one. Okay, a microbiologist is preparing a bacterial growth medium and needs a defined medium. Which of the following component would make the medium undefined? And the options are ammonium sulfate as a nitrogen source, glucose as an energy source, peptone as the nitrogen source, sodium chloride to maintain osmotic balance. So, which component would make the medium undefined but not the defined one? So, what is the right answer for this? Yes, Gyanendra, C is the right answer. So, peptone as the nutrient medium. So, as I told you, for the defined medium, uh, we should have the chemical that have a very specific composition. So, defined medium contain only chemically known compound in the specific amount, and peptide is derived from protein hydrolysis and contain an unknown mixture of amino acid and peptides, and that's why this can make the medium undefined. So that's why C is the right answer. So we have chemically defined medium or undefined medium. Whenever we have medium that have very defined constituent, we know the molecular structure and the degree of purity, then we have defined. And if this is not the case, then this will come under undefined culture medium. Okay, the next question is about the sterilization. Then a hospital is preparing surgical instrument for reuse. For example, if hospital needs to reuse the syringe or the needle, then which of the following method is the most appropriate to ensure complete sterility, so that there should not be any kind of infection? So the options are soaking in alcohol for 30 minutes, washing with detergent and hot water, autoclaving at 121 degrees Celsius under pressure, and wiping with a disinfectant solution. So, what do you think? What is the right answer for this? So, what method of sterilization we will use? Yanendra gave me C answer, and yes, I agree with the C answer that there should be autoclaving. The first answer, the first option is soaking in alcohol. So, this alcohol will not completely kill all the bacteria that we may have on the instrument, and if we want to use that instrument again, then this may be the not the right option. Washing with detergent and hot water, this is not giving us the promise to kill all the bacteria. Then autoclave, yes, autoclave is the one of the promising technique by which we can kill all the organisms, all the bacteria, fungi that we have on that instrument. And wiping with the disinfectant solution, so the role of the disinfectant is to reduce the number of microorganism, but again it is not promising us to kill all the bacteria or fungi or another microorganism. So the only best option is to use the autoclave. And so what do you mean by this autoclave? What happened in the autoclave? And I will tell you shortly. And then this must be at 120 degree Celsius under temperature and under pressure. So let me show you how. Mm, okay, so sterilization have a lot of method. It can be the physical method, it can be the chemical method, it can be the mechanical method. And in the physical method, it can be the dry heat sterilization, it can be moist heat sterilization, it can be radiation sterilization. Chemical, we can use bactericide, we can use the gaseous sterilization, and a lot. But let me show you the you have seen in your laboratory, if you are in the university and if you are taking any course of the biology, microbiology, or even the chemistry course, then you must have seen this kind of autoclaves that you have in your laboratory. And this is nothing, this is just like the pressure cooker, where the steam is given, and under this steam a, uh, um, a particular temperature and particular pressure is given within this within this box, and this will give you a higher steam, and after that all the bacteria, all the microorganism will be killed away. So this is the principle. Here you can see that we are giving the steam supply here, we are giving the steam supply, and inside it we keep all the instrument that we need to be sterilized. So this steam will kill all the bacteria, all the microorganism, and this is how after some time this will give you the very sterilized instrument after a few hours. So this is the basic principle of the autoclaving. Okay, the next question is, which of the following medical item do not always require sterilization before reuse? And we have scalpels, endoscopes, surgical forceps, and hypodermic needles. So which of the instrument so this is just like, if we are using that instrument for the external use, so for the external use we do not need much sterilization, but if that instrument is going inside our body, then we need sterilization for this. So you have to just see which of the instrument that is external is in use but not internal in use. So this is the endoscopes. So endoscopes are the instrument that we need for the external use but for not the internal use, and that's why we do not require sterilization, much sterilization before reuse. So that's why B answer is the right one. So here you can see endoscopes are classified as semi-critical item, means they must be disinfected but not necessarily sterilized, because they do not penetrate sterile body tissues, but other instruments, for example scalpels, surgical forceps, and the hypodermic needles are critical item that must always be sterilized to prevent infection. Okay, coming to the next question. A patient has a minor cut on their hand. Which of the following would be the most appropriate for preventing infection? And the options are hydrogen peroxide, bleach solution, formaldehyde, and detergent solution. So if there is any kind of cut on your hand, so what chemical will be used to prevent the infection? Sonia gave me A answer, that hydrogen peroxide. Yes, I also agree with the A answer, that the answer will be hydrogen peroxide only. Bleach solution is not the it will not prevent the infection, and this will give you more injury on your minor cut. Formaldehyde is formaldehyde is used as the um, sanitizer, but this is again not the most appropriate, and detergent solution is again not the possible preventing solution that we need. So A is the right answer. So okay, so we have like, because the one of the option was bleach, so let me tell you, bleach was not the antiseptic. We need antiseptic on that cut so that we can avoid the infection, but we do not need disinfectant. And we need anti, and there should be you people should know the difference between the antiseptic and disinfection. So antiseptic is the substance that we can apply on the living tissue or on the skin. But if this is the disinfectant, that it we can use on some non living objects, or on some surface or on some instrument. For example, we can use the bleach, but but this bleach will be very toxic for the living tissue, or for the living skin. So the substance that we can apply on the living tissue or the skin, and that would be very less toxic. So for example, hydrogen peroxide and iodine we can use as the antiseptic, that will prevent the infection on the minor cut. Here you can see we have a lot of disinfectant, for example household bleach, then we have ammonium compound, then we also have this hydrogen peroxide gas, plasma, formaldehyde, alcohol, a lot. But in the antiseptic we also have some another chemical that we can use as the antiseptic. So you must know the difference between antiseptic and the disinfectant. Okay, the next question is, a nurse is cleaning a hospital room and needs to clean a microbe from a patient's bedside table. Which of the following should they use? And the options are alcohol based hand sanitizer, iodine solution, hospital grade disinfectant, and antiseptic wipes. So if they want to remove the microbes from the bedside table, so this is table, this is not the living organism, this is non-living thing, non-living surface. And if we want to remove microbe from that non-living object, so what that would be? That would be hand sanitizer, iodine solution, hospital grade disinfectant, or antiseptic wipes. So this would be hospital grade disinfectant. As I told you, sanitizer is used for the uh, living organism, right? And if this is the hand sanitizer, then we need it on the hand to remove the bacteria or to kill the bacteria from our hand. Iodine solution is again the antiseptic but not the disinfectant. Antiseptic wipes is again antiseptic, and that antiseptics are used for the living tissue or the living skin. But if this is the floor or this is the surface of some non-living thing, then we need disinfectant, and we need a hospital grade disinfectant to remove the microbe from bedside table. So C is the right answer. Okay, coming to the next question. Okay, a research lab is working with a virus that can cause severe respiratory illness but does not have available vaccine or treatment. Which biosafety level is required? So this is BSL. So BSL 1, 2, 3, 4. So that laboratory is working on the virus, and that virus is giving you the severe respiratory illness, but and we do not have the vaccine or treatment for that particular virus. Then what kind of biosafety level or BSL we should have in the laboratory? And if you people know that some of the laboratory that we have in India, and this lab, like most of the microbiology laboratory have some tag of the BSL. So either they are following the BSL 1, BSL 2, BSL 3, BSL 4. If you will go to the ISC, then they have the BSL 3, means they can work on the virus. That virus has some of the, let's say, very dangerous cause. But here we are, here the research lab is working with the virus, and that virus doesn't have vaccine. So if we do not have the vaccine, then we need highest level of the biosafety level. So highest is 4, right? But if we do have the vaccine for that virus, then we can go for this BSL-3. But if we do not have vaccine, that we must have BSL-4. So just see here, BSL-1 is low risk to personal and the environment. So our most of the microbiology labs are following the BSL-1. Then we have BSL-2 laboratories. So that laboratory have moderate risk to personal and the environment. Then we have the BSL-3, serious disease for human, animal, and plant, not spread by casual contact. So this will come under BSL-3. And if we are talking about BSL-4, so this is very serious disease for human, animal, or plant, and often untreatable. So if we do not have any vaccine for that, then that bacteria, then the lab should follow the biosafety level 4. So this will give you the answer for this. So, for example, BSL-4 is required for highly dangerous pathogen that pose a lethal risk and lack vaccine, such as Ebola virus. And if you want to work on this Ebola virus, so you can't work in a very general laboratory. You must have the facility in that laboratory that following the BSL-4 level. Okay. And that's why it is always said that you should select the laboratory that have the higher funding, that is following the BSL-3 at least, so that you can work on better dangerous viruses. Anyway, so that we come to the next question. So what is the key difference between BSL-2 and BSL-3 laboratories? And the four options are: BSL-3 labs require the use of HEPA. So HEPA is nothing but the high efficiency particulate air filtered, negative air pressure room. Then BSL-2 require personnel to wear pressurized suit, and BSL-3 labs allow researcher to work with live pathogen on an open bench. And D is BSL-2 labs handle only non-pathogenic microbes. So you have to tell me the difference between BSL-2 and BSL-3. So try to visit at least in BSL-2 and BSL-3 laboratory. Then you will get to know, whenever you are entering, you will feel some air controlled into that room. You can't wear the shoes that you have. You have to cover that shoes or your foot with the polythene bag, so that you do not spread any kind of contamination in that lab. So uh, the difference between BSL-2 and 3 is that 3 require HPEA, means the whole air in that laboratory is in a very controlled manner, and this is not in BSL-2. So that's why A is the right answer. uh, this I already have explained. Okay, so next question is: a new antifungal drug is tested in a lab. Scientists observe that fungal cells stop growing but remain alive. How should this drug be classified? So you have to tell me the name of that, the classification of that drug. That drug is stop growing the fungal cell, but this fungal cell remain alive. So the options are fungistatic, fungicidal, bactericidal, and sporocidal. Can you give me the answer for this? When bacteria cells are still alive, but the growth has been stopped. So what kind of drug we have used? So this will come under fungi static. So static means we have stopped the growth of that fungi, but it doesn't mean that we are killing that fungi, right? If we are killing that fungi, then this would be the fungicidal. Okay, this is for bacteria. If we are killing any bacteria, so this is bactericidal, and if we are killing any spores of the bacteria, or let's say fungi, this will come under the sporocidal. But if we are just resisting the growth of the fungi, then this will be the fungi static. So that's why A is the right answer. So just see here, if after a drug, if after a drug the number of fungi is reducing, then we have used this fungicidal. But if we have inhibit the growth, and a little growth will be here, but the like we can't completely stop the growth. But if we do have a very like slow rated growth, then this will come under the fungi static. Okay, the next question is, a doctor prescribe a medication that kills yeast infection rather than just stopping their growth. Which term best describe this drug? So, for example, if we took a medicine and that medicine is not only stopping the growth, but this is also killing the yeast infection that we had on the skin. So, that classification of drug will come under what? So we have four options: fungistatic, mycostatic, fungicidal, and bacteriostatic. We already have explained in the last question. If we are killing the fungi, so yeast is the fungi, right? So if we are killing the fungi, but we are not stopping their growth, so this will come under fungicidal, right? Fungistatic is stopping the growth, but we are not killing the fungi. Microstatic is again the terminology for the fungi only. So this is very similar to the fungi static. And bacteriostatic is that are stopping the growth of the bacteria. But here we are talking about the yeast infection. So yeast is the one of the fungi. So this will come under the C option. Okay, coming to the next question. Okay, so a sterilization process has a D value of 5 minutes. What does it mean? So we have some terminology of this D value, and the options are: the process kills all microbes in 5 minutes. The next is the process reduces microbial count by 90% every 5 minutes. The process prevents further microbial growth but does not kill microbes. The process eliminates bacterial spores in 5 minutes. So what do you think? What is the right answer? If the sterilization process has a D value of 5 minutes, what does it mean? Can you guess something out of this question? Okay, so this is reducing the microbial count by 90% every 5 minutes. So if this is the case, then the D value will be 5 minutes. So let me show you here. So either it is the 90% or it is the 1 log. So let's say from 10 raised to power 4, we are reducing the bacterial count to 10 raised to power 3. And if it is taking 5 minutes, then the D value is equal to 5. Right? And again, if we are going from 10 raised to power 3 to 10 raised to power 2, so we are reducing the 1 log. And if this reducing of 1 log is taking time, the 5 minute, then the D value will be 5 minute. So D value represent the time needed to reduce the microbial population by 90% or 1 log under specific condition. So this is how we can define a D value is equal to 5 minutes. Right? Is it clear? Okay. So the next question is, if a bacterial population start with this is I think 1 million, so 1 million cell, and undergoes a process, this is not 1 million, this is 10 million I think. So 10 million cell and undergoes a process with the d value of 2 minutes. So it means the one log of the bacterial cell, it has been reduced to one log, and this is taking 2 minutes. How many viable cell remain after 6 minutes? So just see here, this is 10 raised to power 6, right? So this 10 raised to power 6, after 2 minutes, this is changing into 10 raised to power 5 after 2 minutes. Again, after 2 minutes, one log has been reduced, and this is changing your number of bacterial cell to 10 raised to power 4. After 2 minutes, this has been reduced to, your bacterial cell has been reduced to 10 raised to power 3. So after 6 minutes, your bacterial cell has been reduced up to 3 log. So initially we have 10 raised to power 6, but this has been reduced to 10 raised to power 3. And the question was like that only. uh, so you need to tell how many viable cell remain after 6 minutes. So 6 minutes, 10 raised to power 3, and this will be equal to 1000, right? So if you have the d value of 2 minute means one log of the bacterial cell will be reduced in 2 minutes. So the answer is 1000. And just see here, uh, so if the 90% of the bacterial cell are been reduced, so we will have just the 10%. So in either way we can calculate. So 10% of this 10 billion, then 10% of this 1 billion, and 10% of 10,000. So this is how also you can calculate. But the normal, I just saw that you just remove one log. So initially, let's say it was 10 raised to power 6, then after removing one log, it will be 10 raised to power 5, and then 4, and then 3. And this is how you can calculate the number of viable cell that we will have after 6 minutes. Okay, coming to the next question. So a scientist wants to break open bacterial cells to extract their DNA without using chemicals. So which of the following method would be the most effective? Sonification, sonication, then ultraviolet radiation, filtration, and the pasteurization. If the scientists want to extract their DNA without using chemical, so we are not using any chemical, so what method will be the most effective? Okay, so to extract or to break down the bacterial cell, we can use this sonication. So sonication is, we are giving the energy sound to the solution that have the bacterial cell, and what the sonication will do, that will break down the cell membrane of that bacterial cell, and this is how the cell of the bacteria will be broken down, and this will give you then from there you can extract the DNA. So the answer is sonication. So just see here. So for example, this is our suspended cell. We are using this ultrasonic waves, and after that waves, what will happen, this was the cell, and this cell will be broken down, and then this is how, let's say these are our genetic material DNAs, then we can extract this DNA without using any chemical, right? So sonication use ultrasound waves to generate cavitation or the bubble formation that leads to the cell lysis, making it ideal for extracting DNA or protein. So this is how A answer is the right one. Okay. Yes. So coming to the next question, again I have question on d value, or on another. So a scientist test bacterial survival in a boiling water bath and record the time taken to kill all cells. So in d time we had 90% of the reduction of the microorganism, right, in particular time. That would be the d time or d value. But here it is the time taken to kill all cell. So what it would be called as? So this measurement is best described as decimal reduction time, that is d value, thermal death point, thermal death time, and pasteurization time. So what do you think, what is the right answer for this? So how much time we need to kill all cell that we have is known as what? So because this is the time, right, and this is not the d value, this is not the point, but this is time. And pasteurization is very different from pasteurization is we are giving some heat to for a particular time, and that's how the number of bacterial cell will reduce, but this is not killing all the cells. So this is not the under the solution, but this is the right option. So thermal death time is the time that that bacterial cell takes to like scientist takes to kill all the cell using some chemical, right? So the answer will be C. So just see here, and this will be at a particular constant temperature, just keep in mind here. So for example, if we had the temperature T1 and this T1 is taking time 10, let's say 10 minutes to kill all the bacteria, right? If we have the temperature T2, if we are maintaining the temperature T2, it may be that the time taken to kill all the bacteria is 20 minutes. Then if we have, let's say, temperature T3, then to kill all the bacteria we may need 40 minutes. So and here T1 is greater than T2 is greater than T3, and that's why whenever we have more temperature, then there will be more efficiency so that we can kill all the bacteria within a short period. So just keep in mind that thermal death time is the total time need to kill all microbe at the constant temperature. Okay, so this is giving you the C answer, right one. Okay, again there is a question like that only. So a bacterial culture is exposed to increasing temperature. Which measure determine the lowest temperature required to kill all microbe in 10 minutes? So what does it mean? So this is again the thermal death point. So just see here, this is the point, this is not the time, right? So this is the thermal death point refers to the minimum temperature that kill all microorganism within a set time. Okay, coming to the next question. A scientist needs to sterilize a heat sensitive liquid. So for example, we have a liquid and we need to kill all the bacteria that we have in that liquid, but that liquid is sensitive, and we want to kill the bacteria without killing or damaging its essential component. So which method should they choose? So we have autoclaving, filtration, pasteurization, non-ionizing radiation. So just see here, in autoclaving, we have to maintain some temperature that will produce the steam. Okay, Pasteurization also, we have to maintain a particular temperature, and that particular temperature may give you the damaging that essential component, and non-ionizing radiation is not among the option. So the only option that we have is the filtration. So using that filtration, we can remove the bacteria or microbes that we had, and we need to remove it so that the liquid can be sterilized. And because the liquid is heat sensitive, then we can't use any method that is dependent on the temperature, right? So that's why B is the right answer, that is the filtration. So just see here how we can filter it, and after filtering we can also grow the colonies, but this is not our use. The only use we had, the that we just need to remove that microbes, and that we can remove using the filtration. Okay, the next question is on active transport. We already have talked about this, I think, in last or last to last class about active transport and passive transport. But again, we have two questions on that. And which of the following scenario is an example of active transport? The options are oxygen moving into red blood cell from the lungs, sodium ion being pumped out of the cell against their concentration gradient, then water moving into a dehydrated cell via osmosis, and the glucose diffusing into a cell without ATP usage. So what do you think? What is the right answer? You have to give me the example of active transport. So just let me tell you, the oxygen moving into red blood cell from lungs is the passive transport. We do not need any ATP, ATP to utilize. So this is not the right option. B is the right option, that we have to pump out sodium so that we can in some of the nutrient into the cell, and this will uh, in this process we have to utilize ATP, and that ATP will be converted into ADP. Then osmosis is the example of passive transport. This is not the active transport. And if we are not using ATP, then this will become under passive transport but not the active. So active means whenever we are using some energy, we are utilizing some energy to do some kind of processes, so that will come under active transport. So that's why B answer is the only right answer. So just see here, uh, we have to let out the sodium ions so that the potassium ions can come in, and in that exchange we have to utilize ATP. That ATP will be converted into ADP. So this is how whenever we require ATP, then this will come under the active transport. Okay, the next question is again on active transport. So, which of the following is not a characteristics of active transport? And the options are require ATP, move substance from low to high concentration, uses membrane protein, always occur without the use of carrier proteins. What is the right answer for this? So is not the characteristics of active transport. Active transport require ATP, so A is not the answer. Move substance from low to high concentration, this we will talk. Then use membrane protein. If we are using membrane protein, then this is the part of active transport only. Always occur without the use of carrier protein. This is again the confusing, and the right answer is D, that it will always occur without the use of carrier protein. So that's how this will come under the passive transport. And if we are going substance from low to high concentration, so if we are going against the concentration gradient, and that's why this is the active transport. So I think I have given the explanation for the active and passive transport enough. So I think I should move ahead with next question. Okay, so we are talking about the pathogens. So, how you will consider a particular microorganism a pathogen? So, the question is which of the following organism would be considered a pathogen? And the options are a bacterium that lives in the human gut and help digest food, a virus that enter human cell and cause disease, a fungus that decomposes dead organic material in the soil, and microbe that colonize human skin without causing harm. So how you will define that particular organism? We can consider it a pathogen. So for the organism to be pathogen, that should have some host cell. Without host cell it can't survive, right? So it must have host cell, and it can use the machinery of that host cell so that it can do the infection to the host cell, and it will cause the disease. So that's why B is the right answer. So virus that enter human cell and cause disease. So, pathogen is the organism that cause disease, and that's how this will give you the option. Okay, now you have to tell me which of the following statement about the pathogen is incorrect. And the options are all pathogen are parasite. Some pathogen do not require a host to survive. Pathogen can include bacteria, viruses, fungi, protozoa. Some pathogens only cause disease in immunocompromised individuals. So you have to tell me the incorrect statement. So the incorrect statement is some pathogens do not require a host to survive, because every pathogen requires a host to survive. So this is the wrong statement. So B is the right answer. So, all obligate pathogens require a host for survival and reproduction. Which of the following nutrient is essential for all known life form? For example, human, for example, bacteria, fungi. So, what kind of nutrient is essential for all known human form? Options are organic carbon, molecular oxygen, inorganic element like iron and phosphorus, and growth factors such as vitamins. So all organism, irrespective of whether they are aerobic or anaerobic, every organism need inorganic element, for example iron and phosphorus. So the C is the right answer. Okay, the next question is on the classification of microorganism. Uh, so, a microbe isolated from a volcanic vent thrive in extreme heat and derive energy from inorganic compounds. So that particular microbe can survive in the extreme heat, and that can also derive energy from inorganic compound. And so you have what can be inferred about this nutrient requirement? So what kind of microorganism we are talking about? So it depends only on organic nutrient for survival. It does not require inorganic nutrient. It likely utilize the inorganic nutrient like sulfur or iron. It must require oxygen for metabolism. So what do you think, what is the right answer for this? So the right answer for this is that it likely utilize inorganic nutrients like sulfur or iron, because you see now that is deriving energy from inorganic compound. So it require inorganic compounds from where it can derive the energy, right? So it doesn't need oxygen. It may or may not need oxygen, but because it may utilize energy from inorganic nutrients. So let me show you. This will come under. chemolithoautotrophs. So chemolithoautotrophs means that a chemical reaction will happen that will live in the rocks. That's why litho. They will form their food, their self, using the chemical reaction. And that's how they will come under chemolithoautotrophs. So the C answer is the right answer, because this chemolithoautotroph can derive their energy from inorganic compound, for example, sulfur and iron. Okay, next question is on monad equation. So, in a batch culture experiment, if the substance concentration is much greater than the half saturation constant, what happened to the specific growth rate? And the options we have: it decreases as substance concentrate increases. The next is it approaches the maximum specific growth rate. It remains constant regardless of substrate concentration. It drops to zero due to substrate inhibition. So you need to know the relationship between substrate concentration and specific growth rate, provided that substrate concentration is much greater than the half saturation constant. So if we do have the higher substrate concentration, then this specific growth rate should also be higher, right? So the B answer will be right, that it approaches the maximum specific growth rate. So let me tell you the formula for this. So this is the monad equation, where this u and this is the growth rate, or you can say mu. Mu m is the maximum growth rate, maximum specific growth rate, as is the substrate concentration. And this ks is half saturation constant. So if you see, if this k is smaller, if this k is smaller, then what will happen? This mu will be nearly equal to mu m. So this mu m is a maximum specific growth rate. So this is how answer would be as per this equation only. Okay, so next we have one equation. If the bacterial culture follow the equation, and this equation is following the first order reaction, so n is equal to n naught e raised to power mu t. What does the term n node represent? We already have talked about this equation, I think, in first or second live session. So you have to tell me what does this and node represent. So we have four options: the final bacterial population, the rate of growth, the initial number of bacterial cells, and D is the half saturation constant. So what do you think? What is the right answer for this? Can you tell me the right answer for this? You can write in the chat box out of this A, B, C, D. So n node is the initial number of bacterial cell, and we can calculate the final number of bacterial cell if we do know the growth rate, that is mu, and the time taken. And this is how we can follow this equation for the bacterial culture. So C is the right answer, the initial number of bacterial cell. Okay, the next question is on Liebig law of the minimum. So, what do you mean by this Liebig? And based on that, we have the question. So, according to Liebig law of the minimum, the growth of an organism is primarily determined by what? So, options are the most abundant nutrient available. The B is the least available nutrient relative to its requirement. The C is the overall availability of all nutrient, and D is the presence of micronutrients. So what do you think? According to Liebig law of the minimum, the growth of organism is primarily determined by what? The nutrient that is in high concentration, the nutrient that is least available concentration, but and the overall availability of all nutrient, or the presence of micronutrients. This should be B answer actually. Why this is B answer? So see, a Let me show you the graph figure here. So for example, for the growth we need a lot of nutrient. So here you can see there is a pot, and that pot have some calcium, magnesium, nitrogen, potassium, water. So there will be kind of damage or leakage when we have something in the minimum amount and relative to that is required. Okay, so for example, here due to this nitrogen, that nitrogen was in minimum quantity, and because of that minimum quantity, whole system has been disrupted. So according to the law of the minimum, uh, that mainly the growth of that organism is determined uh, by the nutrient that is available in a very low quantity as respect to the quantity that is required. So for example if we needed 10 ml of or 10 mg of nitrogen, but we have just 5 mg, then this will give you the disruption of the growth, right? So that's why and most of the time this is micronutrient. Most of the time uh, the nutrient is micronutrient, but it may or may not be possible. So because micronutrient are always present in the less quantity, but it doesn't mean that the organism also need into in the higher quantity. So that will give you the law of the minimum. But if we have the microorganism and micronutrient and micronutrients we just need in the small quantity, and we have small quantity also, then this will not come under the Liebig law of the minimum. But the nutrient that we have least available relative to its requirements. So that's why not the D answer, but B answer is the right one. So for example if we have the macronutrient as well, so we have for example we have nitrogen, we have phosphorus, and if they are available least available nutrient as comparison to that we need it for the growth, then this will come under the Liebig law of the minimum. So under this Liebig law of the minimum it can be micronutrient as well as the macronutrient. Okay, so that's why B is the answer. So just here see, Liebig law state that the scarcest nutrient relative to the demand is the limiting factor for the growth. Okay, so in an aquatic system, which of the following is most commonly considered as the limiting nutrient for algal growth? So we have four options: nitrogen, phosphorus, carbon, and oxygen. So most of the time in the river body or in any water body, we have some limiting factor, and that inhibit the growth of the algal bloom. But whenever we have that particular nutrient in the sufficient amount, that will give you the blooming of the algal, and this will give you the eutrophication. So this will cover all your water body with the plants, and this is how this will lead to the death of the water body. And so can you tell me what is the limiting nutrient for the algal growth in the water body? So yes, most of the time this is phosphorus. And if you know that you after using that detergent and the water that have the detergent, if that detergent water will go into the water body, that will give you giving you some amount of phosphorus in the water body, and this is how your detergent water can give the algal bloom in the water body. And that's how we always say. And even the fertilizer, if you know the name of the some of the fertilizer that have fertilizer, like most of the composition of the fertilizer is NPK. So NPK means nitrogen, phosphorus, and potassium. So whenever you are applying this fertilizer into your farming land, and this water will move to the water bodies, so water body will have phosphorus, nitrogen, and potassium in the higher amount. And this is how initially the water body has the limited amount of nitrogen phosphorus, but now the water body have a lot of nitrogen and phosphorus, and this is how this is giving you the algal bloom, and that's how there is a death of various of the water bodies just because of the fertilizer that we are using in the farming land, right? And the option is because initially it was the phosphorus limiting water body, but after applying this fertilizer, we are making this water body full of phosphorus. So yes, so that's why B is the right answer. So in freshwater ecosystem, phosphorus is the primary limiting nutrient because its availability control algal growth. Okay, so we have one ratio, and that ratio is known as Redfield ratio. What happens if the P ratio in water is less than 7? So what will happen? Nitrogen is the limiting nutrient, phosphorus is the limiting nutrient, carbon is the limiting nutrient, and no nutrient limitation occurs. So if we do have NP ratio less than 7, then this is nitrogen in the limiting nutrient. Nitrogen is the limiting nutrient. So just see here. This is the red filled ratio where carbon is 106, nitrogen is 16, and phosphorus is 1. But when we have NP ratio less than 7, then nitrogen will be the limiting nutrient because phosphorus is relatively more available. Okay, the next question is, why is the colony count range considered statistically significant between 30 and 300 CFU per plate? So answers are because I show you in one figure, we are doing this dilution series. We have some bacterial colony in after one dilution. If we are doing the second dilution, we have some colonies. After some dilution, we have some colony, and this colony will be reduced as we will undergo this dilution series. But why this 30 to 300 CFU per plate is considered significant? If there is the number of the colony less than 300 or more than 300, why we are just considering that range between 30 and 300? So the options are to prevent error due to too few or too many colonies, because bacteria can only grow within that range, to ensure each plate has the same number of colonies, and D is because larger plate cannot hold more than 300 colonies. So why we just want the number of colonies that have the number between 30 and 300 per plate? Because more than 300, it will be many more colonies to count, and if this is below 30, then we may have some biasness. So we want that much dilution that is giving you the number of the colonies between 30 to 300 only. So that's why A answer is right. So to prevent error due to too few or too many colonies. So the statistical significance of 30 to 300 per plate ensure that too few colonies do not lead to inaccurate estimation, and too many colonies do not cause overlapping, making counting difficult. So that's why A is the right answer. Okay, so we have one more method where for the bacterial culture, and this is pour plate method. And what is the primary reason why pour plate method may lead to heat shock for bacteria? And the options are the use of molten agar at temperature above 45 degrees Celsius, the need for higher incubation temperature, the solidified agar prevent bacterial movement, the colonies are exposed to oxygen which inhibit growth. So why pour plate method may lead to the heat shock for the bacteria? And if it is related to the heat means it is directly related to the temperature, right? So here what we are using the molten agar, and that molten, so to molt that agar, we have to give the temperature above 45 degrees celsius. And if we are giving the temperature above 45 degree celsius to the agar and to then to the bacteria, then bacteria may have this heat shock, and that's how the bacteria can be killed. So A is the right answer. Okay, so coming to the next question. So here you can see this is pour plate method. So first what we have to do is we have to multiply the agar, and then we have to the sample poured onto this sterile phase, and then we have to mix, and then we will incubate. So so this is the method whenever, like when we must have the molten agar. So for that we need the temperature, and that temperature can give the heat shock to the bacteria. Okay, which of the following oxygen species is the most reactive and cause the most damage to microbial cell? And the options are superoxide, peroxide anion, hydroxyl radical, and singlet oxygen. So, which oxygen species is most reactive and can cause the most damage to microbial cell? So, this is hydroxyl radical. So, this is hydroxyl radical that is more dangerous. Okay. Why are reactive oxygen species toxic to microbial cell? What does that species do to the microbial cell that they are dangerous? So the options are: they disrupt DNA and protein, leading to oxidative stress; they help bacteria generate more ATP; they are required for bacterial respiration; they provide bacteria with additional oxygen for survival. And because the question is why they are toxic, so if they are toxic, then this may disrupt some of the functioning. So that's why A is the right answer, that they disrupt DNA and protein, and that may lead to the oxidative stress. So that's why this reactive oxygen species may be toxic to the microbial cell. And here you can see that how they can damage the bacterial cell and can damage it. Okay, so which enzyme is responsible for breaking down hydrogen peroxide into water and oxygen, producing bubble in a positive test? So, for example, if you want to break down this hydrogen peroxide into water and oxygen, you need some enzyme, and because of that some enzyme, that will convert into water and oxygen, and this water and oxygen will give you the bubble in the positive test. So what kind of enzyme we can use to break down this hydrogen peroxide? So options are superoxide dismutase, catalase, peroxidase, superoxide reductase. Janendra gave me D answer, but the catalase is the right answer, because a superoxide oxidase is another class of the oxygen that will react with the that will react with the oxygen species. But this is the catalase that can break down this hydrogen peroxide into water and oxygen. Okay, just see here, catalase break down H2O2 into water and oxygen, but superoxide dimutase will remove superoxide radical, and peroxidase break down H2O2 but do not produce oxygen bubble. And the D option is, if we are using this oxide reductase, that is found to the obligate anaerobic and does not generate oxygen. So that's why B is only the right answer. I will take just 2 minutes more and then we will finish it. Which enzyme allow obligate anaerobic to neutralize superoxide without generating oxygen? Here I think you can give me the answer, that the answer you gave me. So if we are not generating the oxygen, that this may be the superoxide reductase. Okay. If for example, if we need some condition where we need anaerobic condition, so we do not need oxygen in some of the media, then how we can create that anaerobic environment for the bacterial incubation? So we have four options: using a candle inside a jar, placing the bacteria in direct sunlight, adding oxygen to the culture medium, and using high pressure sterilization. So how we can maintain the environment that doesn't have the oxygen? So for that, what we have to do is we have to have a candle inside the jar, because that candle will utilize the oxygen, and then oxygen will be insufficient, and then that will create the anaerobic environment. So A is the right answer. Okay, but this is a very primitive method to create the anaerobic respiration. We can also use anaerobic chamber or glove box uh, over this anaerobic jar for maintaining the strict anaerobic. And why? Because it allow completely removal of oxygen. It may not be possible that candle will consume all the oxygen, but we have very advanced anaerobic chamber and the glow box that can completely allow you the removal of the oxygen and may create the anaerobic condition. So that's why A is the right answer. Then if we do have the chemostat, then the primarily factor to control the microbial growth rate in the chemostat is the inflow rate of fresh medium. Then we have like why we are preferring the chemostat over a batch culture, and this is because it maintain continuous growth and steady state condition. Then again, we have like which of the following best describe commercial sterilization? So if we can destruct the pathogenic and spoilage food in the organism in the food, then that can describe the best commercial sterilization. Then again, we have the difference between antiseptic and disinfection. Then antiseptic is used on the living tissue, while disinfection is used on non-living surfaces. Then which method physically remove microbial rather than killing it? So this is degerming. What can describe best sanitation is that we can reduce the microbial count to safe public health level. Then we can describe that the best sanitization. Then there is a difference between flash pasteurization and the bulk pasteurization, because in flash pasteurization we have to give the temperature but for the shorter period, but for the bulk pasteurization we have to give a lower temperature but for the higher period, and that's how you can have a difference between these two. And the advantage of flash pasteurization is that it alters flavor less and more effective against heat resistance bacteria. Then the true statement about the bulk pasteurization is that it uses the lower temperature but for the longer time. Then if we are using UV light for the microbial control, then it will damage the DNA and DNA of the microbe, and that will lead to the cell death. Then if we are using the irradiation to remove the microorganism for the food, then the major concern is that that can give you the radioactive contamination, and that can also give the alteration in the nutrition value. So there is like mixed reaction whether we should use irradiation methods to remove the microbes from food or not. Then we have the difference between depth filter and membrane filter. So depth filter have non-uniform pores, but membrane filter have uniform pores. Then again, we have difference between bacteriostatics. So static means it can inhibit the growth, and cidal means it will kill the bacterias. So that's why C is the right answer. And bacteriolytic agent, so what they will do, they will lyse, break down the cell of the bacteria, and this is how they will kill the total number of cells. Then we have minimum inhibitory concentration, then the lowest concentration that we need to inhibit the bacterial growth. Why the I think this is the last, second or last, I think. We have the one method is disk diffusion, and in this disk diffusion we measure the zone of inhibition around the antibiotic disk. You can just read about this. And the major reason for tailing in the disinfection curve, you can search for the disinfection curve. And the tailing is just because the disinfection concentration decrease over time, and that's why we have the tail for the disinfection curve. I think this is the last question only. So thank you so much for giving me too much time. And if you have any query or the suggestion, you people are welcome to give. You can write in the chat box, you can like write in the email. My email ID is Jyoti, JYOTI 2121 at the rate iiserb.ac.in. So if you have any problem, you can tell me. If you have any suggestion, you can tell me. Again, we will meet next Monday from 6 to 8 this time only. And till then, thank you so much. Thank you so much for listening so patiently. Uh, Thank you, everyone. Thank you, Anu. Sheikh, Abid, then Gyanindra, Radhika, Alikya. Thank you, everyone. Thank you so much. Thank you, Tanuja.